Hello, good morning, everyone, or good afternoon. I'm going to wait for about a minute just to let more folks log on who have registered, and then we'll begin the webinar shortly. Thank you. Okay, we'll go ahead and get started. Good morning or good afternoon, everyone, and welcome to today's webinar. I'm Dr. Todd Herron from Greenstone Biosciences, and we're thrilled to have you join us today for what promises to be a fascinating discussion on the future of drug discovery. Before we begin, just a few housekeeping notes. This session will be recorded and shared with all registrants after the recording. Please use the Q&A feature to submit your questions throughout the presentation, and we will address them during our dedicated Q&A session at the end. So just a little bit about Greenstone Biosciences before we begin. For those new to Greenstone, let me take a moment to introduce our company and the transformative work that we're doing in the field of new approach methods. Greenstone Biosciences is a computational biotechnology company based here in Stanford Research Park, Palo Alto, founded by Jade Chow and Professor Joseph Wu of Stanford University. We are revolutionizing drug discovery by leveraging cutting-edge human-induced pluripotent stem cell technology, or iPSC technology, that we'll hear more about today from Dr. Mercola. Combined with artificial intelligence and advanced human microphysiological systems, we operate at a critical intersection. FDA regulatory changes are demanding more human-relevant preclinical testing, and technological advancements available at Greenstone Biosciences now makes this possible at scale. Our mission is simple but ambitious: to accelerate drug discovery, reduce reliance on animal testing, and bring safer, more effective therapies to patients faster. We do this through what is called new approach methods, or NAMs, human-relevant models that predict clinical outcomes better than traditional animal models. Our website at greenstonebio.com, you will see our human cell products listed under hiPSC-derived cells. We can generate many different cell types, different human cell types representing many organ systems for preclinical drug discovery, development projects, and customers can select custom cell types and may choose from our over 2,000 unique patient cell lines based on disease, sex, age, ethnicity, and specific gene mutations that cause disease. We can also provide organoids for studies. These aren't just healthy control cells. Our repository includes cell lines from patients with inherited diseases like hypertrophic cardiomyopathy, dilated cardiomyopathy, long QT syndrome, arrhythmogenic right ventricular cardiomyopathy, Brugada syndrome, Duchenne muscular dystrophy, spinal muscular atrophy, and numerous other rare diseases. This diversity is critical because genetic background profoundly influences drug responses. A compound that is safe in one population may cause severe adverse events in another. Our biobank enables pharmaceutical companies to conduct what we call a clinical trial in a dish, testing drug candidates across genetically diverse populations before ever entering in human trials. On our website, you'll also see a list of Greenstone research services ranging from PBMC collection and iPSC reprogramming to custom differentiations and in vitro disease modeling for high-throughput drug discovery. What sets us apart is the integration of artificial intelligence throughout our platform. We don't just generate biological data, we apply computational modeling and machine learning to extract insights, guide candidate selection, and optimize lead molecules. This AI-powered approach dramatically accelerates timelines and reduces costs. As regulatory agencies like the FDA increasingly emphasize alternatives to animal testing, particularly following the FDA Modernization Act, platforms like ours are becoming essential infrastructure for drug development. We're not just replacing animal models, we're providing more predictive, more human-relevant data that improves clinical success rates and patient safety. Please check out our website at greenstonebio.com for more information on Greenstone products and services. Now, we're honored to welcome Dr. Mark Mercola, Professor of Medicine and a leading figure at the Stanford Cardiovascular Institute. Dr. Mercola's pioneering work in stem cell biology and cardiovascular medicine has fundamentally changed how we approach drug development and disease modeling. His lab was instrumental in discovering that Wnt inhibition is critical for cardiogenesis, work that has laid the foundation for producing cardiovascular tissues from pluripotent stem cells at scale. He's collaborated with medicinal chemists, engineers, and software developers to pioneer the use of patient-derived iPSC-cardiomyocytes for disease modeling, safety pharmacology, and drug development. Dr. Mercola's research directly aligns with Greenstone's mission, and his insights will illuminate how iPSC technology and AI are transforming drug discovery from bench to bedside. Without further ado, Dr. Mercola, welcome, and thank you for joining us today.
Thank you very much, Todd, for the generous introduction and, of course, the opportunity to be here. I'm quite honored. Thanks also to Jade and Joe for the invitation and the opportunity. I'll share my screen and start. Let's see here. Do you see the screen?
Sorry, yes, we can see it. It looks great.
Great, good. So I'll be telling you about our efforts to use iPSC-cardiomyocytes to model heart disease, and with a long-term goal of assessing the pathogenicity of genetic variants, in particular correlating in vitro phenotypes with clinical outcomes, and using this information to develop novel therapeutics. So I'm going to tell you about the use of iPS cells and other cardiovascular cell types for drug discovery and development. As I'll discuss, we prepare these cells. I don't think I need to explain iPS cells to this audience, but so just very briefly, we prepare iPS cells from patient donor cells, typically blood cells, and they're reprogrammed in vitro. And then we can drive their differentiation to cardiomyocytes or any other cell type that we know enough about the developmental biology to direct the differentiation. So we direct the differentiation to cardiac cell types, put them in multi-well plates in essence, and we can then use those for high-throughput physiological studies. A key feature of iPS-derived cells that underlie their utility as disease models is that they retain donor genetics. That means for monogenic diseases that act in the cell of interest, say a cardiomyocyte, these cells will recapitulate the disease phenotype in the dish. iPS-derived cardiomyocytes and other cell types have widely been considered as excellent models to facilitate drug discovery, to assess the therapeutic efficacy of investigational molecules, to assess drug safety, and help understand how human genetic variants affect clinical outcomes. A roadblock, however, has been that these studies have often not been performed at scale, nor have they been performed with the depth of phenotyping that allows us to draw conclusions such as, does it correlate with clinical outcomes, or will these drugs have disease-modifying activity in people? And this is largely what we've been devoting our attention to over the last couple of years. So, as I said, iPS cells originate from a cell from the donor, typically a blood cell or a skin cell. You reprogram them and then you expand those cells to create a pluripotent cell that, for all intents and purposes, resembles an embryonic stem cell, a cell that forms the embryo and is present in the conceptus shortly after fertilization. With directed differentiation, you can make any cell type, as I said, and it takes about 3 weeks to make cardiomyocytes. The cardiomyocyte differentiation is now extremely robust, thanks to the efforts of a lot of people. And once you get these cardiomyocytes, however, we must remember that they're terribly immature. So in order to model diseases, it's important to mature the cells so that they have the architecture more closely resembling that of an adult cardiomyocyte. Mind you, they still are very mature, but nonetheless, with some metabolic maturation over a month or two, you are able to tease out disease phenotypes. So I'm going to talk about 4 things today, 4 applications of iPS cardiomyocytes. The first is cardio-oncology. Cardio-oncology is a major problem, as I'll explain, and our efforts have been focused on redesigning oncology drugs to remove the cardiotoxic liabilities. Cardiotoxicity is the second reason that cancer patients die. It often limits the dosing of the cancer drugs, and so it's a very large unmet clinical need and burden. Our second area of interest is repurposing approved drugs to treat rare diseases. So rare diseases are often not profitable for large pharma to take a new chemical entity approach, but there's a lot of room for coming up with drugs to treat rare diseases by repurposing drugs that have already been approved and that can rely either on their on or perhaps their off target effects. Third area that I'll discuss is understanding how genetic variants can influence clinical outcomes. This is really important, not just to be able to say whether a genetic variant is going to be benign or pathogenic, but also because if you can say that the disease phenotype in the dish correlates with a clinical outcome, that means you could use this platform for prognostication. It would also mean by extension that if you use those readouts to discover drugs, those drugs should have disease-modifying activity in people. And finally, I'll tell you about work that's been ongoing and has moved to a company now for heart regeneration. And this involves the development of a biologic. It's a modified form of a naturally occurring adipomyokine that is capable of regenerating the hearts in mice, rats, pigs after myocardial infarction. So I'll focus first on the cardio-oncology. So as I said, cardio-oncology is a major unmet need. It was first recognized many years ago, a couple of generations ago, in response to radiation and anthracyclines. But this problem has continued despite the entry of molecularly targeted therapeutics into the clinic. It was once thought that with very targeted therapeutics, we would have safer oncology drugs, but unfortunately, this has not turned out to be the case. So why is a drug cardiotoxic? Drugs could be cardiotoxic broadly speaking, for 2 reasons. One, on target. The actual relevant target of the drug for the intended indication might be the reason for cardiotoxicity. Maybe it plays an important role in heart function, so inhibition of that target in the heart would be detrimental. Then another reason, perhaps even a more common reason, is off-target problems. The drug might affect a target that's, although not important for its intended indication, nonetheless is inhibited by the drug, and this target might play an important role in the heart. So in order to understand whether the platform could be useful to avoid cardiotoxicity if implemented early on in a pharma pipeline, we selected a drug that for which there's evidence that its cardiotoxicity was off target. And we reasoned that if we could understand the off-target cardiotoxicity, we could engineer the drug so that we would remove its ability to inhibit that target, but retain its ability to inhibit all the targets necessary for its intended indication. The molecule we chose was the oncology drug tyrosine kinase inhibitor ponatinib. Ponatinib is used to treat chronic myeloid leukemia. It's the third generation compound. Imatinib and nilotinib are first and second generation compounds. These are active site inhibitors of the BCR-ABL fusion kinase. The first and second generation molecules are first in line therapy today, but unfortunately, a mutation that blocks entry of these molecules into the active site frequently arises. This is a threonine 315 isoleucine mutation, and it renders the oncogene resistant to these drugs. So therefore, ponatinib was developed because it can get around the isoleucine and get into the active pocket. And it was originally positioned when it was developed as basically a molecule that would be used for all CML patients with BCR-ABL translocations. And however, it turned out to be highly cardiotoxic, was removed from the market, and then re-entered the market with a restricted label. So it's only used with patients that have the T315I, so-called gatekeeper mutation. Ponatinib's toxicity is really extensive. It was noted early on, even in the clinical trials, albeit those were higher doses than used typically today. But as many as a third of people would suffer from some sort of adverse cardiac event. About 6 to 9% of people, even in the original trials, were developing heart failure. So it has a serious risk of myopathy, and in particular of vascular occlusive disease. This graph here shows that with time, where the dosing has been improved, the burden of cardiotoxicity has somewhat declined, but still remains. It persists despite the lower doses of the molecule. So this is a really egregious molecule, and we wanted to see whether we could re-engineer it. And we thought there was hope because the first and second generation molecules aren't nearly as cardiotoxic, so we reasoned it was an off-target effect. As I said, ponatinib was developed a number of years ago by Ariad, which was subsequently acquired by Takeda. Since the first generation and even some of the second are relatively safe, relative to ponatinib anyway, we thought it would be an off-target effect. But there's a big challenge. These kinase inhibitors, active site inhibitors, block about 50 kinases or so at or below the free Cmax values dosed in patients. So, and we have really a very poor idea about which ones are needed for toxicity, for anti-tumor efficacy, and which ones, if different, cause the toxicity. So we couldn't use a directed approach based on knowledge of the inhibitors. And it's even worse than just kinases because there are lots of proteins in the body with ATP binding pockets. And so this molecule could cause toxicity by interacting with one of those. So we had to use then a phenotypic assay, which simply read out the cardiotoxicity. So we assembled a group of talented individuals. This was on my side, on the biology side, led by Anna Hanatiuk, an instructor in the lab, and Arna Bernil, a former postdoc. And it was in collaboration with Sanjay Malhotra, who at the time was at Stanford, but is now a professor at Oregon Health Sciences. And two people in his lab, a chemist, Malesh Pandrala, and a physiologist, Danir Taylor. Also, Yanis Karakakis, with whose lab we collaborate extensively, was also involved in this project. So the project, as I said, was to try to identify chemical moieties on ponatinib that could be modified to avoid the cardiotoxicity. So in the diagram, do you see my point? Here, let me see if I can get a pointer. Laser, there we go. In here, you can see how ponatinib will enter the pocket, but nilotinib will not. And this is the gatekeeper isoleucine in the mutant kinase. So since these two molecules differ in their ability to enter, and hence their ability to kill the mutant tumor cells, but they also differ in cardiotoxicity, we started by creating, basically like cutting and splicing to a non-chemist thinking about it, cutting and splicing the compounds so that we could probe the structure-activity relationship for cardiotoxicity versus on-target anti-tumor efficacy. And so we created about a dozen compounds with each cycle, about 10 or 12 cycles of diversification, synthesis, evaluation, then going back and guiding new syntheses. And in each molecule that was produced was tested in 3 different assays. We tested for oncologic potency in CML tumor lines that Arna had knocked in the T315I mutation. We tested them for vascular toxicity an assay that Anna had developed, and cardiotoxicity, looking at the myopathic effects, so basically detrimental effects on cardiomyocyte contractility, as well as evidence of arrhythmia. So the assays for this, I should take a step back and tell you about the assays. We've spent many years developing high throughput physiological assays to read out myopathy, in other words, contractile dysfunction, and arrhythmia. And this in my lab has largely been the work of these three individuals here: Wes McKeithen, Ricardo Serrano, and Adithan Kandasamy. And we've been doing this also in collaboration with Valo Sciences, who develops the instrumentation that we use. And this is a high throughput kinetic imager. So basically, it's like an imaging plate reader that you'd use for drug screening, but it collects very high frame rate movies, and we can analyze the effect on individual cells or groups of cells with the software that we've developed. So what I'm not going to go through all of the screen, this has all been published, but what I'm going to talk about will be two of the compounds that came out of the screen, known as 36A and 33A. So all molecules, panatinib, nilotinib, 33 and 36, inhibit wild-type CML. However, you can see the big difference between nilotinib and panatinib's killing of these cells in terms of the dose that it takes to kill the cells. 33 and 36 retain the ability to kill the cells, albeit a little bit less effectively than panatinib. Similarly, if we look at vascular if we look at effect on vascular tissue, we can see that compared to panatinib, which has a very aggressively damages the endothelial cells, that we can see that our 33 and 36A are considerably less potent at damaging the endothelial networks that are established. If we look at cardiomyopathic effects, we see the same story. We can see that panatinib is very myopathic in red here, but we can see that 33 and 36A are considerably less myopathic in two different donor IPS lines. And if we look, and that's in terms of contractility, and we see the same story when we look at troponin T release into the media, which is an index of cardiomyopathy and cardiomyocyte damage. So the molecules are a little bit, 33 and 36A are a little bit less potent against the target than panatinib, but they're considerably less potent in terms of cardiotoxicity. So overall, how this plays out, if one takes into account the decreased potency for killing or the decreased potency against the actual kinase inhibition, the molecules are somewhere between about 10 to 20-fold improved over the original panatinib compound. So that's a significant advance. Do these molecules work in vivo? So we gave animals the tumor. We then waited a number of days to get the tumors established, and we started dosing in these various compounds. And what you can see here is you can see the CML tumors growing, and they grow considerably with the vehicle alone treatment, and panatinib will slow the growth and kill the tumor, and so will 33 and 36A. That's just shown here with the tumor explants, but is shown quantitatively by tumor weight and tumor volume here. Most importantly, the 33 and 36A in this setting did not induce the release of cardiac troponins, the index of in vivo cardiotoxicity. So the molecules are effective against the tumors in vivo, retain anti-killing effect, but they do not cause significant cardiotoxicity. So using this platform, we were approached by Nathaniel Gray's lab here at Stanford. Nathaniel Gray has a long history of working on molecular glue degraders. These are heterobifunctional molecules that will bring a target in proximity with the ubiquitin ligase complex. And he was working on a molecular glue degrader for a bromodomain protein, BRD9, that's involved in a number of different cancers and immune disorders. And in these settings, you would like to inhibit that protein. Couple of clinical candidates had been made, tested, had gone into clinical trials, but the clinical trials failed because of cardiotoxicity. So they approached us with a problem of trying to develop a safer version of this molecule. And so in the end, to make a long story short, they developed this molecule called ZZ7. And ZZ7 targets BRD9 on one side of the compound, and on the other side of the compound engages a protein decaf16, which is part of the ubiquitin ligase complex, but it is not present in cardiomyocytes, but present in the target tissues, the tumors, for instance. So we took this molecule, Anna took it, and tested it for problems with cardiac contractility and also evidence of arrhythmia. And you can see here that ZZ7 was very clean, basically, in all of these assays. So we think that this collaboration has led to a novel BRD9 molecular glue degrader that spares cardiomyocytes. So in summary, then, we've used these different tissue types made from iPS cells, cardiomyocytes, vascular endothelial cells, and then we've also used the tumor cells to guide the development of safer oncology drugs, safer than would be developed if you weren't to include this at this early step. And so this is an argument that this sort of assay, iPS cardiomyocyte safety assays, could be implemented in the pharma pipeline very early, at the point of hit to lead. The typical pharma paradigm for cancer drug development is that human studies for safety, or animal studies for safety rather, are rather late in the program, after a drug candidate, or at the point of where the drug candidate is being nominated. I think that it would be much better to implement these studies using human cells, not animal cells, early on in the process. I think this would increase the predictivity of toxicity because the cells are human and a more realistic model, and it would allow the structure of the molecule to be adapted early on while there is still time before a lot of expense is devoted to the project to take the molecule into animals or worse, into clinic. So I think that we could address a large part of cardiotoxicity prior to entry of these molecules into clinical development. So what did we do about molecules cardiotoxicity when it isn't an off-target effect, but it might be an on-target problem? Can we develop a mitigating strategy for on-target cardiotoxicity? So Anna has initiated a screen looking for protective targets. In other words, proteins in a cell that, if inhibited, would protect against the cardiotoxic effects. And she posed as the cardiotoxic molecule, panotinib, because of our experience with that compound. And then she screened siRNAs for their protective effect. And in the end, this screen resulted in 3 different targets. And these targets were, interestingly, alone, if you inhibited them, had no effect on cardiomyocyte contractility, but they would mitigate the contractile deficits caused by panotinib. Furthermore, they would also mitigate the problems caused by doxorubicin, which is really exciting because the mechanism, of course, is completely different. So we think we have a few interesting targets to explore. We've since gone on and just begun in vivo studies with one of these genes for which there is a clinically approved inhibitor. And so at this point, this is just emerging data, fresh off the shelf. The experiment was just terminated a week ago. So we have echocardiography data throughout this time frame, but we've dosed animals with panotinib and the improved inhibitor of one of these protective targets. And we're following survival and cardiac function. I don't have the cardiac data at this point, but hot off the press is the survival curve, which looks really encouraging. So the treatment is able to significantly protect the animal's viability. So we're very encouraged by this study. We might have a clinically proved molecule that could be used to protect against cardiotoxicity of panotinib, perhaps of other molecules as well. Okay, so I'd like to switch gears now and turn to repurposing approved drugs to treat rare diseases. And there's a big need for being able to come up with new drugs for heart failure. We have relatively few therapeutic strategies, many drugs, but they target relatively few therapeutic strategies. But they've been really fairly successful. And it's been argued, as even a decade ago by Joe Loscalzo, that perhaps our success is actually in some way creating a conservatism towards that prevents us from exploring completely novel strategies. Another reason for conservatism is that conventional drug discovery for cardiology, as all of you know, is very costly. Costs can exceed $2.5 billion per drug. Consequently, there are very few new drugs, only 1 to 2 FDA approvals per year for the last 8 or so years. And that's out of about 50 approvals each year for different indications. And most of those approvals are not for completely novel drugs. So there's a big need to come up with to improve the early stage of drug discovery to present new targets and new chemical matter for drug development. And this, I think, would be revolutionary for patients because it would allow us to treat the underlying mechanisms of the disease rather than the consequences of the disease. And it might also personalize medicine so that depending on the particular mechanism, you would now have a very focused armamentarium, much like cancer therapy. So our focus has been on different myopathies, and here for this project, I'll focus on dilated myopathy. It's the #1 etiology for heart failure, for heart transplantation. It's highly genetic. There are many genes that are known to cause, that contribute to dilated cardiomyopathy. Many are monogenic, and current therapies address symptoms, and although they do slow progression, the underlying pathogenic mechanisms are poorly understood, and these drugs are not really focused on these mechanisms. So using our platform, I think it should be possible to develop, to identify new drugs, either by understanding the mechanisms and starting drug discovery de novo, targeting those mechanisms, or looking or repurposing and hoping basically to get lucky. Many diverse mutations give rise to DCM, as shown on this slide. I'm going to focus for this talk on just one mechanism, that regulates calcium cycling from the sarcoplasmic reticulum into the cytoplasm, which directs contractility. And the DCM, genetic inherited form of DCM I'm going to talk about, is due to a mutation in the phospholamban protein, which is a negative regulator of the SERCA2 calcium reuptake pump. And that's shown in a little more detail here. So calcium is sequestered in the SR because SERCA is pumping it up with each cardiac cycle, released through the ryanodine receptor, where it can then activate calcium influx from the outside the cell, and also, of course, triggers contractility. And then as the contraction ceases, the calcium is taken back up into the SR. So this mutation is actually it arose in an individual in the Netherlands. It's the phospholamban R14 deletion. It arose in the Netherlands about 700 years ago in the north of the Netherlands. And in fact, many of the carriers of this mutation that are known, there are about 1,500 known carriers in the Netherlands, reside basically surrounding, with the epicenter being where the disease probably originated. But however, the Dutch, of course, have traveled all over the world historically in the last 700 years. And so you find hotspots of this mutation in the northern Midwest of the United States, the Midwest of Canada. You find it in Spain, you find it in Asia, you find it in many places where the Dutch have traded or fought. And so it basically is a map of the Dutch presence in the world. The disease basically is highly arrhythmogenic. It's a highly arrhythmogenic dilated myopathy. It resembles in many ways arrhythmogenic right ventricular myopathy. You get a fibrous fatty deposition. Patients often present with arrhythmias, and they need to be transplanted. The presentations typically occur when a patient is 30s or 40s, and then the symptoms tend to worsen over time. And about by old age, about 70–80% of people have some form of the disease. Carriers, about 30 or 40 years old, I think the penetrance is about 30% or so. So it's a serious disease. And it's one for which there's a fair amount of interest on the part of the patients. They're very well organized in the Netherlands. There's also a US PLN Foundation. And there have been many efforts to try to figure out what we could do to to mitigate this disease. Of course, a curative therapy would be a gene therapy. that would remove the mutant allele. There are many individuals working on that. A very elegant therapy would be to rewrite the genetic code in the mutated allele. There are a lot of efforts towards that, but these are far off. And so we've been focusing on trying to find near-term solutions to the problem in anticipation eventually of having a genetic solution, or gene therapy solution. We've worked in the space here of looking at molecules that would activate the unfolded protein response, which is pathologically upregulated, but we found that if you stimulate it further, you can actually protect against the disease. But what I'm going to tell you about is an unbiased repurposing effort that has actually focused our efforts on in this area here on the calcium cycling, and also a focused effort on CAM kinase. So taking a step back, we developed iPS cells for the phospholamban disease by two ways. One was to take cells from our first donor, patient number 1, is Peter Glynas. He's the founder and director of the phospholamban mutation. He was transplanted about a decade ago. Here he is looking through the microscope in our laboratory at cardiomyocytes made from his cells. So he's looking at his heart cells, but of course he does not have his own heart cells in his body anymore. We've made a corrected version of those to make the isogenic control. We've also taken healthy individuals and we've introduced the mutation. This was all done by postdocs in the lab with the help of Yannis Karakaitis. So we can recapitulate the contractile deficits in the dish. So you can see the patient relative to the corrected version. This patient cells generate less force. Similarly, conversely, introducing the mutation also leads to less force generation. Other hallmarks of the disease are an upregulation of the UPR that we can see in vitro. We don't actually see much of an arrhythmic phenotype in vitro, probably reflecting the reductionist monoculture model. But nonetheless, the contractility, kinetics of contractility and the amplitude are good readouts of this disease. So we reasoned that empagliflozin might be a reasonable therapeutic for this disease. Empagliflozin is a sodium glucose transporter 2 inhibitor developed to treat diabetes. It was approved for heart failure with both reduced and preserved ejection fraction a number of years ago, but it all has an off-target effect against CAM kinase 2 delta. And so because of the elevated diastolic calcium in this disease and activation of CAM kinase, we thought that this might be an interesting therapeutic target. And indeed, if you treat cells with empagliflozin and you look at just the contractile traces, you can see a normalizing effect just on representative traces, and that's quantified here. You can see an effect in 2 different patient cell lines, or in the introduced line as well as in the patient line. If we look at calcium kinetics, the main feature is that the downstroke of the calcium transient is slowed, as you would expect if there's increased diastolic calcium in the patient lines. And empagliflozin will speed up, in essence, the downstroke of the calcium transient. And you can see that here if you look at the change in calcium decay times, so that we can see a decrease in the time it takes for the calcium transient to decay. So empagliflozin might be one molecule that could be used in patients. We also took an unbiased approach. The empagliflozin data has been published. This is an unpublished study in which we used an approach that was popularized about a decade ago from the Broad Institute and at NIH. And in this approach, what you do is you look at a transcript profile for your disease. So basically your biological state, the disease, has a transcript profile. And you're looking at databases of drug effects on transcriptome profiles. And you're looking for an inverse correlation to identify drugs that would reverse, in essence, the transcriptomic signature of a disease. And so you basically have your query, which is your disease. You compare it to the reference database, and you look basically for drugs that would cause a reversion of the phenotype, a strong negative outcome from this comparison. This is a really elegant strategy, although in actual practice, its performance has been somewhat limited. There have been a few statistical studies of it and that have highlighted shortcomings, and in particular, a discordance between the different results you get with different reference databases. And there are also issues that the cell, and this might be in part linked to issues that the drug databases of transcriptomic effects are from cells that don't relate to those of the disease state itself. They're often tumor cells or immune cells, but they're not going to be cells of disease interest to us anyway, cardiomyocytes or to many others. So it has been somewhat limited, but nonetheless, it's a very elegant and potentially powerful approach. So we started this by establishing a collaboration with an informatician at the University of Cincinnati, Anil Yega. And we took differentially expressed genes in our iPS-cardiomyocytes from the patient. We looked at differentially expressed genes in the right ventricle of the mouse and in the left ventricle of the mouse, and fed these then into the connectivity map algorithm. And what this does is you are then able to look at these are individual drug effects, and you can look at drugs that either reverse the signature, you can identify drugs that mimic the signature, but we focused on those that are the best at reversing the signature. And if you look at signature reversal in the different data sets, different transcriptomic sets that we used, you can see the different number of drugs we found or reference compounds we found. And we focused on these here that give substantial overlap with the iPS cells. So we tested those in assays. I'm not going to go through all of the data with testing all of these compounds. We tested them for contraction amplitude, relaxation rates, calcium transient kinetics, and so on. And what we found at the end of this is two molecules looked particularly interesting. And I'm just showing you the contraction amplitude results, but same effect is seen with relaxation kinetics. What you can see is a restoration of both of these two compounds restore the contraction amplitude and contraction relaxation kinetics relative to the isogenic control. Interestingly, they appear to, although they rescue contractile defects, they do nothing to the calcium transients. So we believe they're working somewhere between calcium and the effect on the sarcomere, possibly to modulate calcium resistance, calcium sensitivity of contraction. So they're interesting compounds. Compound A is quite a benign molecule. These are both approved drugs, and we have actually dosed a first patient with compound A because it was ethically reasonable to do so in the Netherlands. And so we're watching this patient, and we hope to initiate a clinical trial with compound A. I'd like to make a transition now to understanding how genetic variants can influence clinical outcomes. So this is a project that's been led by David Stout, a pediatric cardiologist in the lab. He's particularly interested in restrictive myopathy, which is a disease in which the ventricular myocardium stiffens. And so these patients have notable diastolic dysfunction. In pediatric inherited forms of restrictive cardiomyopathy are quite serious. Patients often need to be transplanted in the first few years of life. Diastolic dysfunction also exists in hypertrophic myopathy, of course, where the muscle is basically bulked up. And that's opposite to the situation you see in dilated cardiomyopathy, which is largely a disease of the systolic function, in other words, the squeeze of the cells. So with the help of Ricardo Serrano, the engineer who developed the software for this, and Peter Tran, a medical student who was in the labs now actually graduated, they performed a really interesting project to survey different mutations in troponin I3 and look at their effects on diastolic and systolic function and correlate those effects with clinical outcomes. So how do we measure diastolic dysfunction? So we use a form of traction force microscopy in which beads are embedded into an acrylamide gel on the bottom of the plates, and the cells are then plated on top of those, and they cause the deformation of the underlying substratum, and we track that by looking at the beads. So we can see then you can generate basically traction forces showing the divergence of the beads, and because we know the material properties of the substratum, we can calculate then the force that's exerted. And we can calculate the systolic force, in other words, the squeeze, but we can also, by comparing it to the the gel without the cells, the position without the cells, we can determine the amount of tension at rest, in other words, during diastole. So we get diastolic as well as systolic forces. So how do you, so then we wanted to screen many troponin I3 gene mutations. Major problem is that cardiomyocytes made from iPS cells are very immature. They don't even make much troponin I3, but they have troponin I1. So what we did is developed a platform in which we deleted both troponin I1 and troponin I3 from the iPS cells and introduced and made a double knockout line and introduced into those by lentiviral transduction either wild type or HCM or DCM variants of troponin I3. This way, we would be able to use the double knockout human iPS platform to read out the effect of troponin I3 variants at scale. So when you do this, what you see is that the wild type cells give contraction traces like this. The level here at the bottom would be the diastolic tension. The difference between the bottom and the top would be the systolic tension that's generated. Interestingly, RCM and HCM variants tended to have a very high diastolic floor, a very higher systolic ceiling as well. But note that the difference between the two is less. So that's consistent with clinical data. DCM, on the other, is the opposite effect. You see a decreased floor, a decreased ceiling, and a decreased net systolic force. GFP alone in the system, of course, does nothing. So armed with this platform, we wanted then to overlay calcium because we wanted to understand the relationship between calcium and contractile effects. So here's a video of the field of view with the beads. You can see the beads moving with contraction. You can see the green flash for the calcium probe. So we overlay bead motion with calcium flash in these simultaneous acquisitions. And so you can get calcium and force traces. And from that, you can see then that you can easily discriminate wild type contractility from HCM, RCM, and DCM. And this using a metric for calcium duration, for in essence, calcium sensitivity. It's the contractile duration normalized by the calcium duration. So it gives, that is a metric then of the sensitivity of contraction. So you can see that DCM, you can see a decreased sensitivity, and HCM and RCM variants in troponin I, you can see an increased sensitivity. So if we screen across the variants, what you see is that you get a really nice segregation of the DCM, RCM from the synonymous or control variants, and then you can see the DCM down here. It's a perfect segregation. We can easily call the difference between, for pathogenic variants in this model. We can easily determine which variants are pathogenic by looking at the difference from the synonymous or the controls or the benign. So an issue here is we collect lots of metrics. So in addition to looking at calcium sensitivity, the instrumentation, the software that Ricardo developed can spit out a tremendous number of metrics, about 30 individual metrics, which can be combined. So we wanted to know which of these metrics would be good for segregating the different types of HCM and RCM causing variants into different groups to give us some kind of basically phenotypic resolution to put them along a scale. So David then created a family of random forest classifiers for pathogenicity using all the parameters and combinations. And he ranked those by importance, basically for the ability to classify, like how well do they classify? And he ended up with 5 metrics, and they're shown here. And these metrics allow the segregation of the phenotypes into different clusters. So they clustered into wild type benign, a couple of DCM clusters, and 3 different HCM clusters. And if we focus on the HCM/RCM clusters, you can see that they segregate neatly in a principal component plot. And here's how the different metrics, and I'm not going to belabor this, but the different metrics will discriminate the different classes. Individual metrics will discriminate the different classes. So the real point of doing this is what we want to know is whether these variants, the in vitro phenotype will look like will correlate with real world clinical outcomes. So for that, we turned to the Stanford Center for Inherited Diseases, and a fellow there, Brendan Floyd, worked with David to pull out the patient data. This is a very consistently phenotyped cohort of patients. And we asked for those that are carrying the mutations that we have tested, can we see, do we get a correlation with one of our metrics with disease severity? And the metric that worked out the best turned out to actually be the age of onset. And the metric for the in vivo metric, and the in vitro metric that worked out well was basically the extent of diastolic dysfunction. So it's diastolic tension as a fraction of maximal tension. So how high is the floor relative to the overall tension that's made? And what you find is that these variants will correlate then with the variants allow the correlation of the in vitro and the in vivo phenotypes. So we got a good correlation with age of onset. This is, well, there are 2 points I'd like to make of this. Firstly, it's really interesting that calcium sensitivity itself, which so nicely segregated the pathogenicity of variants, didn't correlate with the age of onset. And we speculate that this might be because increased calcium sensitivity is needed to get the disease, but it might not dictate the degree of diastolic function that causes actual clinical presentation of the disease, that influences the clinical presentation, but rather the actual measurement of diastolic tension correlates better, we believe, with the diastolic disease dysfunction that then brings about the clinical severity. Okay, so what I've told you here is that we have sorry for the typo, scalable multi-parametric platform for analyzing the functional consequence of troponin I variants. We can easily discriminate pathogenic from non-pathogenic. We come up with these very complex multi-parametric measurements that nicely split variants into functional subgroups, and that these analysis of patient data revealed that these subgroups correlate, and that suggests possible clinical relevance. To our knowledge, the importance of this, I really want to emphasize this. To our knowledge, this is the first demonstration that in vitro physiological phenotyping correlates with clinical outcomes. And it was made possible not only by the consistently phenotyped patient cohort, but importantly from a technological standpoint, the depth of the measurements and the scale of the study. So it's important not just for being able to say one variant will give you a certain risk of presenting early in clinic, but it also means, and for drug discovery, that if we were to use these metrics that correlate with clinical outcomes, that drug effects against those parameters will reveal drugs with disease-modifying activity in people. So I think this is a very important step forward for the field. It has certain limitations, of course, as shown here, and we're trying to address these as we move forward. So I'd like to conclude in the next few minutes with a discussion of heart regeneration and the development of a biologic to regenerate heart tissue after myocardial infarction. This is a project that's been going on in the laboratory of my lab and that of collaborator Pilar Ruiz Lozano, who's also my wife. This came out of her laboratory and was part of her insight. Our contribution to it was developing an assay that allowed the identification of the substance. So the concept was that the epicardium is important for fetal heart development. Therefore, the fetal epicardium might produce molecules that could be used to regenerate the adult heart after myocardial infarction. And so Pilar's logic was to take fetal epicardial condition media and ask whether it would have an activity that would promote the replication of ES cells in the days when this was started, ES-derived cardiomyocytes that we could quantify by the presence of a green fluorescent protein. And what she found was that firstly, the conditioned media, and then later, after mass spec identification and sifting through molecules, that one protein in the conditioned media, follistatin-like 1, would promote the replication of immature cardiomyocytes in this setting. And remarkably, if this was put on an engineered collagen patch, and sutured over the surface of a mouse infarction, you could see that over time, that animal will recover its ejection fraction or the extent of fractional shortening. It took a few months to recover. So that correlated with a decreased scar volume. This was data that was published in Nature about actually a decade ago now. And so it decreased scar volume and restored cardiac function after MI. This was a really remarkable result for us. And what was interesting about this at a biochemical level, oh, I should take a step back. Before I get there, the regeneration of myocytes occurred because pre-existing myocytes were creating the new muscularized tissue. And this was shown by an in vivo lineage labeling in which you pulse basically all the pre-existing cardiomyocytes so that they are green with a Cree system. And now you remove that pulse and you do it, in essence, a chase with the regeneration. And you find that the new myocytes created are also green. And they've undergone at least one round of replication at some point. They've incorporated EDU. So one of the most exciting aspects of this was that the native human FSTL1, which is expressed at high levels after injury, does not have this effect. What the active molecule had to be non-glycosylated, or in the case of the fetal epicardial form, hypoglycosylated. So the extent of N-linked glycosylation affected whether FSTL1 was cardioprotective, which had been shown before, versus regenerative. Okay? And what's particularly cool about this is that zebrafish, which can regenerate their hearts, have the same patterns of N-linked glycosylation sites as does human FSTL1. And that's shown in the lower left here. And so there are 3 sites for N-linked glycosylation. And interestingly, in zebrafish as well as in human, in the zebrafish A, but zebrafish B, which is the one that comes up after injury, has only 2. And when you delete zebrafish B, but not zebrafish A, now the fish after injury will form a scar. So zebrafish B, with its lack of an N-linked glycosylation site, is important for promoting regeneration in the fish. So we find that N-linked glycosylation is important in the mammalian in vitro setting and in vivo setting for regeneration, and it seems to be important in the zebrafish. So functional study was done by Lior Zangi in New York, in which he individually mutated the asparaginins that are N-linked glycosylated in FSTL1. He mutated them each individually and all 3 together. And regenerative activity was only seen when he introduced these by modified RNA into an infarcted animal. He only saw regeneration when either the first site was mutated or all 3 were mutated. That's the same one, sorry for the orientation being flipped, but it's the same one that's mutated that's missing in the zebrafish. So that asparagine or mutating all 3 renders the protein regenerative, just like what we found when we used the non-glycosylated form of FSTL1 manufactured in bacteria. So here I'm going to present new data that shows that this will work not just in healthy animals that have been infarcted. It's one thing to take a mouse or a pig and infarct the animal, a juvenile animal, you infarct it, you then come back and treat it right away. That's very different from the intended patient population, which might have comorbidities, might have multivessel disease. And so it's very important to ask the question whether this therapeutic will work in a setting of comorbidity. So what we did was we used pigs, Ossabaw pigs, which are predisposed to becoming obese and developing metabolic syndrome and diabetes. So we fed these pigs basically a fat-rich Western diet. So they were on McDonald's for 4 weeks, basically, or 4 months rather. And then they were infarcted. We waited 30 days, and then we started administering Reg-101, which is the non-glycosylated form of the protein, subcutaneously. So this is a major difference over the earlier studies where we used a patch to do it. Now we're administering the protein subcutaneously, which is much more translatable towards clinic. So it was administered for 2 weeks, and then the animals were left to recover for another 2 weeks, and at termination, the hearts were examined. And if we look just grossly at the sections, you can see there's a decrease in the necrotic area that's quantified here. We've always been interested in why FSTL1 or non-glycosylated FSTL1 has no effect on healthy myocardium, and nor does it have an effect on adult cardiomyocytes. They must be immature. And so we looked at the proliferation in the remote area. We did not see that. That's not shown here. We also looked at the changes in energetics. And what you could see in the remote area, there's no change in the energetics of the myocardium, but there is a substantial change in mitochondrial respiration in the border zone of the infarct. And so this, again, is consistent with our earlier results that we only see proliferation of cardiomyocytes and remuscularization within the border region of the infarct, even when the protein now is given subcutaneously. If we look at cardiac function, we see evidence for a recovery of systolic and diastolic recovery. This is by invasive hemodynamics at the end of it. We see a number of other parameters also improving, in particular, the proportionate pulse pressure. So this is in essence proportionate to the head of the pressure head that's going to perfuse tissue. It's a very important metric of heart failure outcomes. Heart rate goes down as the animals, and we know they're less congested. Heart rate is going down, indicating less sympathetic response. So the animals are feeling better and they're performing better. We note that Reg-101, or the hypoglycosylated, non-glycosylated FSTL1, does 3 things that are really important as predictors of death and hospitalization. One is it decreases scar size, increases ejection fraction, and reduces volumes. And these metrics are very important, have been shown in meta-studies of clinical data to support as being important to predict reduced adverse cardiac events such as rehospitalization. And these studies are shown here. So since Reg-101's effect size is a couple-fold larger than what was seen by the sacubitril and valsartan combination in the paradigm trial, we believe that the reg effects are very likely to translate into clinic, just as Entresto has done. So in summary then of the overall talk, I've told you about 4 applications of iPS-cardiomyocytes. One is in cardio-oncology, where iPS cardiomyocytes and vascular endothelial assays supported the development of safer versions of 2 oncology drugs. A safer version of ponatinib and a novel BRD9 molecular glue degrader. We've also used the cells to repurpose drugs for rare diseases, and we have 2 approved drugs that we think will be useful and could be positioned to ameliorate the contractile deficits and maybe the arrhythmic deficits of phospholamban cardiomyopathy. I've also told you that in vitro phenotypes of pathogenic variants of troponin I3 correlate with clinical outcomes, in particular, namely age of onset. And this is important not just for disambiguating variants, but also suggests that if we look for drugs that modulate these parameters, those drugs might have disease-modifying activity in patients, giving us a clear path from in vitro to clinic. And finally, I've told you about heart regeneration, where FSTL1 lacking N-glycosylation functions distinctly from the native protein, acts in the border region to restore functional myocardium, and the effect is apparent not only in healthy animals that we've given an infarct to, but also in older, sick animals that have type 2 diabetes as a comorbidity, similar to our intended patient population. So thank you very much. I'll stop there and take any questions.
All right. Thank you very much, Dr. Mercola, for that comprehensive and inspiring presentation. We've received a few questions in the Q&A. Before we get to those, I have one question that I'd like to ask regarding the phospholamban study. So thinking to traditional animal-based cardiomyocyte studies for a protein defective in calcium signaling like phospholamban, A lot of experiments to be done would be looking at, for example, calcium sparks and other types of diastolic dysfunction that could happen and contribute to the disease. In your lab, or is there any evidence in the literature of measuring calcium sparks using iPS-cardiomyocytes?
Yes, Don Burr, not in our laboratory, no. But Don Burrs has been looking at this in particular with respect to phospholamban. And those are ongoing experiments, as I understand it. So I don't really want to comment on them. But I think just taking a step back, the the contractile deficits are very apparent in the phospholamban disease. But as I said, the arrhythmogenicity of the disease is not so clear in the cardiomyocytes. We do see elevated diastolic calcium. We see a delay in the downstroke, as I mentioned, but translating that actually to arrhythmia has been challenging, not just for us, but in general in the field. But this is where work like what you're suggesting would be important.
All right, thank you. So I'll get to some questions from the Q&A session. If anyone has questions, please feel free to write them into the Q&A window. One comment is about the chemotherapy part of your talk. So this says vasculogenesis is a way the tumor uses to grow. Maybe the effect on new vessel formation is a drug effect that you want.
Quite right. I mean, what we were hoping for here is simply an assay for how healthy the endothelial cells are. So I do agree, though, with the question that if you were looking for a drug, you might want a drug that impairs endothelial function in the tumor, but spares it in the body. I don't know how to achieve that. I know, though, however, there is a caveat to the very good question, though, that there is in some tumors, at least, people feel that reconstructing the vascular perfusion of the tissue would actually aid in chemotherapy. I'm not enough of an expert to comment on that, but the question is very good, and I think it's a complex question. We're using that assay simply to read out an adverse effect on the vascular endothelial cells.
Okay. Another question relates to siRNAs to block cardiotoxicity. The question is, are they cardiac specific? And there is the risk to minimize the effects of the drug also on tumor cells.
Okay, very good questions. The genes themselves that we're not thinking about using the siRNAs or shRNA as a therapy, but we really would like to have a small molecule or other systemically delivered drug to target those genes, their protein products. In the case of the three that I mentioned, no, they are not selective to cardiomyocytes. Therefore, any of the drugs or means of inhibiting those could themselves have off-target effects. That's why, but we have taken one of them forward into animals because it has minimal adverse effects when used. Secondly, it's actually might contribute to an anti-cancer effect for the tumor. I don't want to say too much more about it, and because I really am not in a position to divulge that molecule just yet.
All right. Thank you. Another question from the Q&A says, what % of drugs developed can reach heart tissue? And then second question is the dose increased so that it can reach heart tissue, or do drugs readily reach the heart?
Very good questions. Do drugs generally reach the heart? I believe the answer is yes. I'm going to have to punt on that or speculate on that question, though, because I'm trying to think of bioavailability studies for different drugs. Individual drugs I know of reach the heart, but I do not have a good estimate of, in general, do drugs reach the heart? Or are there ways conventional ways of modifying small molecules to keep them away from the heart. I'm not aware of that, like there is for the brain, of course. But targeting drugs to the heart is actually a very active area of research. Getting that selectivity is interesting. But the opposite, how do you keep a drug out of the heart? That's a very good question. I don't have a good answer. I'm sorry.
Right. Another question says, thank you for the informative seminar. Could you please share the system you use to measure the contractility of hiPSC-CMs in your cardio-oncology studies?
Sure, glad to. The platform itself is an instrument that is made and sold by Vala Sciences in San Diego. And the software that we use to score contractility was produced in the laboratory. We've described that in the literature. I'm glad to actually share the software, so please contact me and we'll share the code. We have an engineering publication on it, but we're preparing now a biologist-friendly publication describing the software and its use.
All right, thank you. That's very generous to offer the use of that software. Another question says, can the TNNT assay be scalable to other sarcomeric and non-sarcomeric variants to adjudicate the pathogenicity?
Yes, it was troponin TNNI3. The system is in principle scalable. However, there is a limitation that we used a lentivirus to introduce the variants into the knockout chassis. And the lentivirus can only accommodate a certain size protein. So it'd be harder to do that with larger proteins, but that would be the limitation. You could switch to adeno or another way to introduce the mutated genes. But yes, it'd be scalable. We're not the first to have used a knockout iPS-cardiomyocyte as a chassis to introduce others. This was done by Hinson at Jackson Labs in Cincinnati. I believe he was one of the first to do this. So we did not come up with that concept, but it was essential to make the double knockout because of the troponin I1 that was present in the cells.
All right, thank you. Another question.
It's also aided, I should point out, that it's particular. This approach is well suited to sarcomeric proteins because you always get a nice, if you overexpress the protein, you get a nice stoichiometric integration of sarcomeric proteins into the sarcomere. It might not be the case if you were to move away from the sarcomere and look at other structures. We also did a lot of upfront work to very carefully titer the viruses that were used to get a consistent protein expression, even though at the end of the day, I think the cardiomyocytes would incorporate the proteins stoichiometrically. But it was we put a lot of time and effort into delivering the library under conditions that produce comparable amounts of protein across the library.
All right. Another question is, do iPS cardiomyocytes develop disease mimicking structural phenotypes? For example, myocyte disarray in HCM or sarcomere rarefaction in DCM?
Do they? Yeah. Yes, with caveats. I mean, it depends on the extent. The maturation, I think, is really the problem here. That to see these structural correlates of the disease, the cells need to be matured for a considerable amount of time, and then they have been scored. It's challenging to do that in the high throughput setting, and for practical reasons. So the answer is, I think, an equivocal yes.
Mm-hmm. All right, thank you. So I'm just going to go through two more questions for the sake of time. So the next question is regarding TNNI3 variants. Did you try to perform single-cell RNA sequencing of iPS cardiomyocytes to dissect them?
Yes, we did. And in the context of the screen. And that's another arm of the analysis. That's a completely different arm of the analysis. The paper describing that project is under review now for the physiological analysis. The RNA-seq analysis is basically that arm of the project is a distinct. We're treating it as a distinct project. I think it's fascinating in and of its own right, and so that's underway. But yes, and it's all of these were barcoded, so it's easy to tease things to link back the single cell seq with the variants. Yes.
Okay, and just one last question. It starts off with a compliment saying this was a great presentation. Thank you so much for sharing with all of us. Does the therapy that induces cardiac regeneration still work in older, non-juvenile pigs where the endogenous repair mechanisms might not be as potent?
That's very good. That is a really important question. So that's what the point. These are older pigs, and they do have diabetes and metabolic syndrome as a comorbidity. So we were trying to mimic our patient population, intended patient population. But I think that's will be something that awaits. A real answer to that is going to await clinical development of the project.
All right, so that concludes the Q&A session. And in closing, Dr. Mercola, I just want to thank you on behalf of everyone at Greenstone Biosciences and all of our attendees today for sharing your expertise, for sharing unpublished data, which is always exciting to see, and your perspective on the use of iPSCs for personalized medicine and for the use of new approach methods to replace animal studies where possible. As a reminder to all the attendees, this webinar has been recorded and will be available to attendees as well as being posted on our website. Thank you all again for joining us, and I hope everyone has a great day and a very happy holiday season. Thank you.
Thank you very much, Todd.